Okay. But so what I thought I would do today was, I'm as you just heard, a clinician, as a scientist, and I thought I'd give you a bit of a tour through some of the research we've been doing over the last few years to try and come up with some new therapies to treat this condition of heart failure. Okay, and it's a little bit of a cook's tour. I've sort of started to bring in some different concepts. So really what I'm going to talk about is some new drug therapies, and these drugs maybe make you live longer. So that's the Immortality that we'll talk about. We're then going to talk about a new technique in cardiac MRI, which really utilizes the principles of quantum mechanics and physics to move it forward. And then I'll talk a little bit about regenerative cell therapy or stem cell therapies there. So hopefully I don't put you all to sleep. There is Coke, Diet Coke at the back. But what I think we should do is, if you have any questions as we're going along, just please put your hand up, throw some pizza at me, yell out, whatever is required. Can you adjust that? Okay. Excellent. Okay, so I think everyone's settling in. So hopefully this video's play. Yeah, no. So obviously what my favorite organ in the body is the heart. And so this is just a little schema to remind us all what we're focusing on today. A little bit of a fly through. So now we're entering in the pulmonary veins, and we're entering the chambers of the heart, the atria. There's the bicuspid or mitral valve, 2 leaflets, and now we're entering the left ventricle, and we're looking back up at the aortic valve, which you'll see the 3 leaflets there, and these funny hand-like things are the papillary muscles, and this is, if you like, the journey of the red blood cell as it gets ejected out the valve, and we're looking back on the aortic valve again, and as we enter the aorta. Now obviously the heart's important, it's a pump, so its job is to squeeze and push the blood throughout the body, and it does so in order to meet the metabolic demands of the body. Okay, so the heart's job is to get blood where it's required, which is your muscles, because when your muscles contract, that is then needed to actually allow us to move, to think, to walk, to eat, to do every little component that we require of in life. And I'm not a very smart person, so what I did is I thought I should take a quote from someone who was smart. So this is going back, this is a French philosopher, Voltaire, back in the 1750s, and he said, "The art of medicine consists in amusing the patient while nature cures the disease." So as a little statistic for you, it wasn't until penicillin was discovered in the 1930s that when you went to see a doctor, that you had a greater than 50-50 chance that that doctor would actually do more harm than good. So up until the 30s, when you saw a doctor, you could flip a coin. It may actually make you worse rather than make you better. And it wasn't really until the antibiotic penicillin came into play that we were then able to give it to our patients who had infections and improve their outcomes. So that's not that long ago, 1930. And it's interesting, the story about penicillin when it was first discovered, someone made the observation that when they had some fungi on the petri dish when they were trying to grow some bacteria, that the fungi prevented the bacteria from forming bacterial colony forming units, smart enough to then take that observation and realize, oh, maybe it's producing something that inhibits bacterial growth. So what they did is they then purified it, and that's penicillin. Now the first person who ever received penicillin was a policeman who was injured, and he had a nasty infection called Staphylococcus aureus, and this infection was traveling through the bloodstream. So they took all the penicillin they had and they injected into the policeman, and the policeman started to get better. So they realized, here we go, we've only got one person, but the drug is doing something good. Unfortunately, they didn't have enough penicillin. So the infection came back and the policeman died. But it was proof of concept that actually this observation works. So that's the 1930s, and as you know, since then, we have now many cures for many different areas of medicine. And so what I thought we'd do today is focus on a little bit about a big and emerging problem that we face in society. And that issue really is the problem of heart failure. Okay, so what is heart failure? Heart failure is a syndrome where the heart is unable to pump enough blood to meet those metabolic demands of the body, okay? You're not getting enough blood to the muscles to enable them to work effectively. And it's a big problem. So you'll see at the moment there's around 600,000 Canadians living with heart failure. There's about 50,000 new cases diagnosed every year. Almost 1 in 2 Canadians will know someone or are going to meet someone who will develop heart failure, and it's expensive. So in Canada, we're looking at around $3 billion a year. I think by 2030, it'll be about $16 billion a year. And in the US, they're predicting by 2030, it's going to be around $800 billion to treat this. So this is an expensive problem, and we just don't really have the resources to be able to treat it. So what we've got to try and do is come up with new discoveries to prevent it. Now, unfortunately, it's a bad issue. So if you admit it, so I work on the cardiology ward service here at St. Michael's Hospital. If you're admitted to hospital with heart failure, let's say it's a young person who's got an infection of the heart or an elderly person who's got heart disease, and you're admitted to hospital, your chance of dying, so death, we're not talking about having a heart attack or something else, we're talking about death, is 16% within 30 days of leaving hospital, and it's 25% at the end of 1 year. 50% chance of dying in 5 years. So this is a bad issue to have. And unfortunately, if you go back and look at 1997, if you're admitted to hospital in Ontario with heart failure, your chance of dying in 1 year was 26%. If we go forward to 2017, your chance of dying if you're admitted to hospital is 24.8%. So yeah, the number's smaller, but when I'm sitting there with a patient in front of me, I say, you've got a 1 in 4 chance of dying in 1997. You had a 1 in 4 chance of dying in 2007. 2017, it's still 1 in 4. So there's actually very few cancers that have the same likelihood that you will die as a result. And unfortunately, we know with a lot of people who survive their cancer, what do they die of? They develop heart failure and die of heart failure. So this is becoming a bigger and bigger and bigger problem. And one of the big drivers of heart failure is you're more likely to develop it the older you are. And of course, we're now in a situation in Ontario where there are more people over the age of 65 than there are under the age of 65. We have an aging population, so we are going to see more heart failure as a result. So why does the heart start to fail? Well, a couple of things happen to the heart that ultimately result in its inability to pump blood out properly. And remember, when a pump, it's got 2 phases. You've got to squeeze the blood out, and then you've got to suck the blood back in. So it's not just all about how the heart pumps. And here's a little video here running where you can see the heart's pumping, and then the heart muscle was thickening up. And one of the findings that we commonly see as the heart starts to fail is the heart muscle becomes thicker. Okay, it starts to undergo this process known as hypertrophy. Now this is important, and I'll tell you why. Some of the organs in the body have the ability to regenerate. Okay, so you know if you lose your hair, what will happen? You'll regrow new hair cells. The lining of your stomach will regrow constantly. Muscles can regenerate. Fat cells generally don't regenerate that much, but the ability of some organs is amazing how much it can regenerate. And I'll talk about that when we talk about stem cells. The heart is not one of those organs that can regenerate itself. Okay, so heart muscle, these muscle cells that you've got here, we say are what is called terminally differentiated. That is, you're born with a certain number of heart cells. Probably within your lifetime, maybe you can recreate, you can double it. You can probably get 1 extra reproduction of some of those little heart cells. But what that means is if they start to die off, ultimately it's going to continue to die off. It'll put more strain on the surrounding heart muscle cells, and that the heart function will get worse and worse and worse over time. So the heart doesn't have a great ability to just create more muscle. It doesn't work that way. So therefore, what we've really got to do is protect it as long as you can, because it's very difficult for the heart to regenerate itself. So we need to make certain that it never develops any injury, and in that way it can continue to contract and relax and do its function as a pump. The other common finding we see in the heart, and the picture that you can see on your right there, is this is a stain in the heart. So we've taken a biopsy. This is actually from an animal there, and have I got a pointer? Yep, here we go. And you can see here, this red stuff is scar tissue that is being deposited in between the yellow stuff here. These are heart muscle cells. So these are the cardiomyocytes there. And in between that, we see the red stuff. And the red stuff is collagen. Okay? So collagen is what makes up scar tissue. So when you cut yourself on the skin and it heals and creates a scar, it does so because the body lays down collagen in that area. Now, unfortunately, the heart does so when it gets damaged because there's not much else it can do. It can't make new cells. It can increase the size of the cells, and that's what creates this process of muscle thickening or hypertrophy. But the only other function it can really do is it can lay down scar tissue because there's not much else it can do. Now, why is this important? So remember I said the heart is a pump. It's got to squeeze blood out, and it also has to relax. Now, when the heart has a whole lot of collagen through it, collagen itself has the same tensile strength as steel. That means it's basically like you're laying down a whole lot of steel cables throughout the heart. So you can imagine the heart here, it's trying to contract and relax, it's contract and relax, but what happens when the heart becomes full of steel? It can contract, but it's harder to do so, and it has a lot of trouble in relaxing. Okay, and why is this important? Because if it has trouble with relaxing, it doesn't fill properly. And if it doesn't fill properly, it can't eject the blood out properly. And if it can't fill properly, the blood's still coming in from the veins, entering the lungs, and it tends to become congested in the lungs. So you develop what's called pulmonary congestion, and therefore you become short of breath whenever you walk. So the heart, remember, it's not only about the contraction, it's about the relaxation, and this scarring becomes a ubiquitous process that occurs. And it doesn't really matter how you damage the heart muscle. You could have a heart attack, which is still the commonest way in which heart failure occurs in society today. Okay? The arteries develop atherosclerosis, they block off, the heart muscle dies because it doesn't receive any oxygen, and that accounts for about 2/3 of all cases of heart failure. So standard heart attack, and that's important because there are 5 typical risk factors for heart disease. High blood pressure, high cholesterol, smoking cigarettes, a family history, and diabetes. So we can treat most of those things. You can't change your family history, but we can stop people smoking, we can treat their blood pressure, treat their cholesterol, and treat their diabetes. So you can prevent that. About 1/3 of the population develops heart failure for other reasons. They may have been born with a genetic abnormality, there may be other reasons to it. That's a little bit harder to treat, but regardless of how the heart starts to fail, the result is the same. Because the heart can't generate new cells, it will thicken up and it will develop scar tissue, and that is important because now we know what's happening. Hopefully we can come up with ways to treat it, so to try and reverse the changes that occur. But if I play the video again, you'll sort of understand one of the problems we have when we're trying to fix the heart. Now remember, the heart's job is a pump. It's got to pump continuously. If the heart stops pumping, what happens? We die, okay? There's a problem. So when we have to fix the heart with our drugs, it's a bit like the car driving down the 401 to go to Montreal, hopefully have fun on the family day weekend, and you're trying to fix the car while you're driving at 120 kilometers an hour because you need to make certain the heart can never stop. You can't just take it out of the chest, we'll put this on here, we'll fix that. You have to try and fix it while it's pumping continuously. And that's why we think this syndrome of heart failure has such a high mortality rate, why it's such a problem, because the heart never gets a chance to arrest, never gets an opportunity like a muscle to heal itself, or maybe your liver might get a chance to regenerate itself because you can go to sleep, let everything relax. The heart continually has to function. And so once the problem occurs, it's a bit of a one-way street. It gets worse and worse and worse over time. And that's why we think those mortality rates, the death rate from heart failure, are such a big issue. So we've been trying to come up with a couple of different ways over the last few years to figure out how to fix the heart. And remember I said the heart itself develops scar tissue over time, and it became pretty clear to me that when I started looking at a lot of my patients, I noted that the same sort of scar tissue that was occurring in the heart we could see here in the face and in other organs. So as you age, one of the things that happens in the organs is you lose your ability to regenerate those organs and you get more scar tissue. So this is the sort of person who's gone through their journey in life, and if you like, the same sort of changes we see in the skin to tell us that the skin's getting older and changing are occurring within the heart. And it just so happens that there are enzymes throughout the body, and one of them is known as this SIRT1. It doesn't really matter what the name of it is, but what this particular enzyme does is it floats around the body and it modifies proteins and DNA to try and remove the hallmarks of aging. And as cells get older, the DNA changes. They accumulate a whole lot of little extra proteins and changes on the DNA, and the proteins themselves get older because the ability to repair is lost. And what was very interesting is we know that as you get older, this particular enzyme here becomes less and less and less able to perform its job. So we thought, well, wouldn't it be interesting then if we could take a drug to improve the activity of this particular enzyme? So this then gets back to the immortal part of the talk, that if we could activate those protective enzymes and stop the aging process, we may be able to stop the scarring, the hallmarks of aging that occur. And it just so happens that there is a drug called this one here, SRT 1720, that activates this particular enzyme. So as you get older, the enzyme's less able to work. We can take a drug to make it back again. So if you like, we can turn you back into youth, the holy grail again. Now there's an interesting thing. This drug was designed based off this particular drug here called resveratrol. Now you might have heard a bit about resveratrol. It got a lot of media mentions because it's present in red wine. So people thought, excellent, okay, the French, they smoke, they eat a lot of fatty food, but they live longer. Maybe it's the resveratrol in the red wine that's protecting them. The bad news is, to get enough resveratrol to have any effect, you have to drink 40 bottles of red wine every day. Okay? So I'm an Australian, I like my red wine. This is still a problem, right? 40 bottles of red wine. We've tried to concentrate it, we tried to come up with all these different ways. Didn't really work, okay? So a very smart guy who was also an Australian came up with this particular compound here, SRT 1720. And just to give you an idea how much interest it is, when he developed this particular compound, he sold it to one of the large pharmaceutical companies for $880 million, right? So that's a pretty good payday for a few years in the lab there. So very interested in this drug. And then what we decided to do was, I'm trying to tell you that heart failure is a bad thing. How can we improve it? Chatting to one of our colleagues up at the Toronto General Hospital, Phyllis Billier, who's an expert in heart failure, and she helps in the transplantation team. So you see Dr. Billier at the end of your journey with heart failure, when your heart's totally failed and the only options you've got is to have a transplantation. What I'm trying to do is put Dr. Billier out of business, to make certain that no one develops heart failure. But what she has access to is, unfortunately, when they do a transplant, they can take a sample of tissue for me. So then I can study it. And sure enough, this is here known as a Western blot, which is a way that we can measure protein levels in the heart. And here's this particular enzyme of interest, SIRT1. And here it is in our controls, okay? So these were actually two women who were in a motor vehicle accident in California, okay? And they were killed during the accident, and they had signed a form to release that their heart could be used for scientific uses. So you're able to purchase a little bit of protein, and we're able to compare that to our heart failure patients. And you can see here, this dark band here is the intensity, how much protein's there. So you can see in our heart failure patients compared to our healthy controls, there's less of it. And this bar graph here gives you an idea. So as you get older and as the heart starts to fail, you would have thought you want more of this protective enzyme, but we're actually seeing less there. So we then decided, okay, so tells us that maybe we're onto something here. So we went back and thought, okay, we can't just take this drug and put it straight into humans. We have to have more data before we can say that it's safe, and that my theory that loss of this enzyme is important. So we went back, and this is where animal research comes into play. My daughter is in grade 12, and she told me that not everyone agrees with animal research. I think that's great. Everyone's allowed to have their own opinions, but this is one way in which we try to identify where signaling pathways occur so that we can come up with the right drugs and move them into humans safely and know that we're on the right track. It's not perfect, but it's one way to go about it. And what we did is we take these mice and we tie off the aorta. Okay, so here's the brachiocephalic trunk, the common carotid artery, the left subclavian artery. We take a little ligature around it there. It's 27 gauge, so it's about 0.1, 0.2 of a millimetre, and we tie it off, and then the heart now has to generate and squeeze blood out at a much higher blood pressure. So it's a model, if you like, of hypertension, high blood pressure. And what happens to the hearts? Well, they do what we saw from the human studies. They thicken up, hypertrophy, and they develop the scar tissue, the fibrosis there. And this is what it looks like here. So this is after we've taken that animal and we've followed it and it's sacrificed, we remove these hearts, we do all these stains to show there's more scar tissue. The cells are thicker in size, and then they get and this is a marker here to look at how many cells that produce the collagen are increased. So we know then that even though it's a mouse, it develops the same sorts of changes that we see with humans. And so what we did is we took this particular mouse and we gave it the drug. So the model is called transverse aortic constriction, so we call it TAC. And what we showed is that when you perform this on the animals, their hearts thicken up, they get more scar tissue, and that when you give the drug, after you've tied the artery off, you're able to prevent all those changes. So you can restore it. There's a lot of extra detail that we went in before then, but basically we also showed that there's less scar tissue in the heart here, and we showed exactly how the drug works. So this is interesting because this tells us that maybe this strategy is important, and it's even more interesting is as we've gone to look at other conditions. Now I apologise for putting this slide up. This is a bit unsightly when you're having your lunch. This is a liver. This is someone who's drank too much alcohol in their life and developed a condition called cirrhosis of the liver. So the alcohol is toxic to the liver cells and damages them. This is someone's lungs, and the reason it's discoloured is they worked in the coal mines their whole life. So they breathed in the coal and it damaged the lungs. And you can see these large spaces in here. That's because the scar tissue, those little alveoli, the little oxygen exchanging sectors within the lungs, broke down, and so you develop these larger air spaces here. But you can imagine if I don't have the pictures here, but if we section these, the liver, the lung is just full of scar tissue. And then if we look over here, this is someone who has a condition called keloid scarring. So instead of, I said when you cut yourself, you have a little bit of collagen being laid down, it causes the scar tissue to form. This is when that process occurs, but for some reason it goes out of control and you have developed these enormous scars. So we've looked in a number of these other different conditions and shown that the drug works in a similar way. It also works on the kidney as well. So in each of these different conditions, you see a reduction in that protective enzyme and that we can restore it. So we're looking to move that forward and hopefully get it into humans and see whether or not it works. But there's still a fair bit more work to be done. So the conclusion of this is scarring, fibrosis is a major problem of the heart. We don't actually have a lot of therapies at the moment. Most of the drugs we give don't specifically target the scarring. And what we think we need to do is not only do you have to help the muscle cells, you have to reduce the scar tissue in order to improve heart function there. So that's the first bit. Now, what I thought we'd do next is talk a little bit more and get a little bit more into quantum mechanics. Now we're going to do a bit of a deep dive into physics, those that are interested in that. So you heard before that cardiac magnetic resonance imaging is the best technique we've got to non-invasively look at the heart. So non-invasive means we don't have to put a tube into anyone. You can lie down, we scan the heart, and we can develop these beautiful looking images here. So this is the left ventricle. So this dark stuff here is the muscle of the left ventricle. Here's the blood pool. Here is the mitral valve. This is the right ventricular muscle and the right ventricle. Here's the tricuspid valve. This is the right atrium, and this is the left atrium. So you can see we can develop these very nice images there. Yeah, question?
What happens when the mitral valve prolapse?
Oh, okay, so we can talk a little bit about that. When the mitral valve prolapses, what happens is you can see here each time the heart contracts, that the valve should snap shut and create a watertight seal. In mitral valve prolapse, what you generally have is an abnormality in the connective tissue. That fibrous scarring that should keep the valve strong, it becomes a little weaker and it develops like an elastic band. And instead of that snapping shut tightly, it bows back and it can create a little gap. And of course that means the blood, instead of getting ejected out the aorta, because the heart's a pump, it should flow in one direction, it can be regurgitate into the left atrium there. So that's what mitral valve prolapse, and it's an abnormality of the scar tissue, the collagen tissue, because we all need scar throughout our body. We all need some collagen to give us the structure to walk, just like you need a skeleton, you need scar tissue. It's a problem when you get too much though, okay? And so this is what we do with an MRI. We take the heart, we look at it in multiple different planes, and over here, instead of what we've done, instead of looking at like here, where we've gone from the apex of the heart to the base, we've now turned it into multiple different donuts. And you can see these are the sorts of donuts here. This is the left ventricle, the muscle there, and then we can measure this. So we can actually go through and do quantitative measurements. So it's a very accurate way to look at how large the heart is. How much blood is getting ejected out in each beat? How well is it relaxing there? But one of the issues that I mentioned before is the scar tissue and the hypertrophy that occurs. Well, it generally occurs later in the disease. Because I said, it doesn't matter how you get there, this is generally the response. So one of the things that we became interested in, well, is there a way to look earlier in the disease, right? Because we want to try and stop the disease before it happens. And one of the things we found was that the heart is, as I said, it's a very metabolically active organ. It's got to contract and relax, contract and relax, contract and relax repeatedly. And that means the heart muscle must be very active metabolically. So we started to think, well, are there ways that we can measure metabolism in the heart? And remember I said, this is where it gets tricky, because you can't just stop the heart to measure it. You need to come up with a technique to measure that while it's continuously moving. And so for a number of years we've been working on this. Now you'll probably be a bit surprised to know that the heart, in order just to continually contract and relax each day, requires to generate about 6 kilograms of the building block of life. Right? That's not 6 grams. This is 6 kilograms of ATP. Just you sitting here, you don't have to do anything. You don't have to go for a run. You don't have to walk back to school or whatever is required after this, just to be able to contract and relax normally. And here's a little schema here. I don't know if you remember your physiology here. There's a myosin, a thick filament with a bilobed head here. This is the thin actin thin filament wrapped around with tropomyosin, this blue filament, and on the end of it there is the hormone molecules. And essentially what happens is the heart interacts, and you can see that sort of ratcheting process occurs, and that's how the heart contracts and relaxes. But each time it does that, it has to use ATP, say the building block of life. If you don't have ATP, the heart will stop beating and you'll die, and you've got to generate 6 kilos a day. Okay? Unfortunately, the heart's only so big, you can't store 6 kilograms anywhere. So there's no ability for the heart to store it. So if it stops pumping, then very quickly, if it's not getting enough oxygen through, it will deplete its stores of ATP. And that's why when you have a cardiac arrest, it's such a sudden phenomenon. You die so quickly because the heart's been beating, and then all of a sudden it stops beating. It very rapidly uses any remaining ATP, and there's nothing left over to contract and relax again, right? So that's why you have to restore heart function very, very quickly, otherwise you can't do much. And in order to generate this ATP, the heart is what we call a metabolic omnivore. So remember the omnivores? You've got herbivores, carnivores, omnivores, eat anything. Well, the heart will pretty much take anything you can throw at it, and it will burn it to generate the ATP there. Because as I said, it's a pump, it can never stop beating. And we've been interested in, for a number of years, in coming up with this particular technique to try and measure it. So long and short, MRI, probably you heard about this early on because of a technique called magnetic resonance spectroscopy. And what that was very sort of instrumental in drug development and understanding the structure of things. Because everything in the universe resonates at a certain frequency. And with MRI, we can send a little signal in, if you like, we can ping it and then listen to the frequency that it resonates at. Okay, and we can use that to recreate an image. And this is exactly what we've done here. So this is a mouse heart. So we've sent our little signal in, we've perturbed the way protons in the heart spin, and then we can listen to the signal and we can use that to generate an image there. Now the reason you can do this, and the reason you can get those nice images, is protons exist in H2O, right? What's water? H2O, predominantly protons, and the body is about 70% water. So there's a lot of protons floating around. So you can send a signal in, and what it means actually, for about every million protons, we can detect the signal from one of those. Now it just so happens, because there's so much water in the body, the sum total of all those signals allows us to take that and recreate the image here. However, when we're looking at the structure of these things that are involved in metabolism, these are about 10,000-fold less abundant in the heart. So they're very difficult to measure. So for the last 10 years, we've come up with a technique to enable us to increase the signal of these metabolic components, glucose, fats, and able to then use that to identify changes in metabolism. So what do? So this is, if you've got a spare $50 million, you can buy a machine like this. This is a $3 million machine, and what it is, it's a magnetic resonance machine, okay? And it's got a microwave in it, and attached to it we have a cooling device. And what we do is we take what are known as MRI active nuclei. So an MR active nuclei is a nuclei like carbon, but it must have an unequal number of protons and neutrons. It doesn't work when you have equal numbers, because basically the charges neutralize themselves. And then what we do is we take this, the MR active nuclei, and you can use lots of different ones. We just use C13 because most of the things we want to measure involve carbon. And we dope it with what's called an electron doped solution. Now, if you remember your Bohr-Rutherford model of valence electrons, etc., what we're able to do, we put it back in this machine and we cool it down. Now, we really cool it down. So as you know, the absolute, 0 degrees Kelvin is about minus 274 degrees. So it's pretty cold. We cool this down to 1.3 degrees Kelvin. So about minus 272.4 degrees using liquid helium. And then we put it in a magnet, and then we send a microwave beam through it. And what it does is it actually transfers the energy in these electrons into the nucleus. So what that basically means is our carbon atom, which before was very low abundance and it was hard to measure, it's a bit like we supercharge it. And we increase the ability to measure that, not 10,000-fold, not 20,000-fold, not 100,000-fold, but about 400,000-fold greater. So now we've actually got something we can measure. And the thing that we actually measure is a sugar that's very important in the heart called pyruvate. So most of the stuff that generates the energy that your heart requires comes from this molecule pyruvate. If you remember your old biochemistry, I know this is where it gets really boring when most people fall asleep. You know that you can undergo anaerobic metabolism to generate lactate. It can generate ethanol from that, or it can be what's called decarboxylated and into the Krebs cycle or citric acid cycle and generate what's called acetyl-CoA. And acetyl-CoA is the stuff that generates the most ATP. So that's why this is important. Pyruvate, I'll show you the structure of it. It happens to have 2 carbons in it. So what we can do, instead of taking our normal carbon, which has atomic mass of 12, we can actually put one of those C13 ones in, those MR active nuclei. And now what we can do is get it ready to inject it and follow how it gets metabolized within the heart, okay? Now, there's a few other steps that went along the way to be able to get it ready for human use, because we had to warm it up. You can't inject people with stuff that's minus 272 degrees. Right? I don't think any of you would like me if I came at you with some liquid helium there and tried to inject you with, it's even colder than it is outside today. So we had to figure out a way to warm it up, but what we can do is we can warm it up fairly quickly to 37 degrees. The pH is 7.4, the same pH within the body, and now we've got something that we can inject it. It only took 7 years for us to come up with that technique, and about another $6 million. And so this is what it looks like. So now what we've done is we've taken that pyruvate, it's this hyperpolarized stuff, it's this fancy new stuff, we've increased the signal, we've injected, this is a pig. So we've taken the pig and we inject it, and what you'll see here is it enters the blood pool and it goes red. Then when it's metabolized, it generates bicarbonate and lactate. So this is actually live metabolism occurring. And we can play around with this a number of different ways so that we can look at what happens in that Krebs cycle as well as what happens with glucose oxidation. Because the way the heart works is you generally burn fats or glucose to generate all that ATP. So now we've got a technique to do it, and in 2016, this is the image on a journal. This is one of the graduate students who volunteered to be the first person to have this stuff injected, and we can see here, this is the yellow, is the pyruvate being injected, and then over here you can see it's being metabolized to one of the byproducts. So now we've got a technique that we can actually use to look at metabolism in the human heart. And the good thing about MRI is there's no radiation involved. So there's no harm to the patient. It only adds about 2 minutes on to the scan, so you can do this fairly quickly. The problem is it cost me about $12,500 per sample at the moment. So it's very expensive, so it's only a research tool, but now we've actually got something that we can do to move forward. And you know, the heart's important, but let's not forget about the brain. Now the brain also uses predominantly glucose, so we've gone on and done a number of scans, and this is example of the first brain to be scanned with this technique. And what we can see here, all the yellow is lactate in the brain, and over here we see the bicarbonate signal now. So this technique, even though we focused on the heart, we can use this to look at the 3 most metabolically active organs in the body: the brain, the heart, and the kidneys, right? And you can use the same thing to inject and measure all those. So now we've got a way that we can move forward. So hopefully we're actually starting to look at patients with diabetes who have lots of problems with heart failure, and patients who have had heart failure, to see if we can understand, is there a change in their metabolism? And as you heard at the beginning, I do a lot of work with diabetes and diabetes companies, and we have developed a number of drugs that we can then modify different parts of the metabolic pathways. So hopefully we'll have something there. Now the last little bit, we just thought a couple more slides, talk about stem cells here. So we generally use the word regenerative cells, and we stay away from the term stem cells. And the reason is when stem cells first came out, and as you know, stem cells were discovered here in Toronto. Tilla McCulloch, so famous Toronto-based discovery. And stem cells, generally people got confused. So stem cell is a cell that can reproduce itself, and there's different types of stem cells. But some people took stem cells from embryos, so embryonic stem cells, and obviously there's ethical issues about that. Well, the cells that I'm going to be talking about we call regenerative cells, because they're actually your own stem cells that exist within your body, and we can take those out and play around with them and get them ready to inject them back in. But the concept of the ability to regenerate organs has been around a long time. So I don't know if it projects very well, but this is an old picture, many years old, and this is Prometheus. Okay, so this is going back to Greek mythology. So Prometheus gave the gift of fire to mankind, and Zeus was very unhappy with him. He said mankind should not have fire. They haven't earned it. So what did he do? He cast Prometheus down to earth and tied him to a rock. And each day he sent an eagle, a huge eagle, would fly in, rip open his liver because Prometheus was tied to the rock, and feast on his liver, and each night the liver would heal itself. This is how the legend goes. Now the reason we use this when we talk about regenerative cells is I think there's a reason that this legend came about, because people understood that the liver actually has a remarkable ability to regenerate. Remember I said the heart, terminally differentiated, it can't, but the liver has a remarkable ability to differentiate its cell and regenerate new cells. And it just so happens that there are lots of stem cells circulating around. In each of us, we have them, and they predominantly exist in the bone marrow. So the big sites of the bone marrow, the long bones, pelvis, sternum here. So these stem cells in various different forms exist in there. So we actually have a source that we can use to identify those stem cells. So we became interested in that, and again, this is going back into animals, and we're interested in one of the big issues we find in patients with diabetes is they develop kidney failure and heart failure, right? So diabetes affects lots of different things. So what we did here is we took some animals, and you can see here, this is a normal glomerulus. So the glomerulus is the little filtration unit of the kidney, and as it develops diabetes, it becomes impaired. So this brown stage here is the normal lining of cells, the endothelium, and this becomes impaired as animals get sicker and sicker and sicker. And so what we did is we took these stem cells that we took from the animals themselves, cultured them up in a petri dish, and then we re-delivered them. And so these animals have had the same injury. Their blood pressures are very high. Their kidney function was very bad when they started, but when you deliver the cells, what we're able to show is that these stem cells can go to the kidney and reverse all the damage that occurs there. Now this is important because when we think about most drugs, what they do is they slow down the progression of the disease, but the disease still continues. What we're talking about with these regenerative cells is actually reversing the damage that's occurred. So it's not just holding things at bay, it's actually trying to reverse it. And sure enough, when we looked at how these drugs actually worked, we went back to our scar tissue again and found that what they actually did is they turned off the process of scar tissue formation. And by turning it off, it allowed the cells then to come in and recreate new cells to allow the kidney and the heart to then function normally. So it truly was a reversal of the process and not just slowing it down. And so at the moment we're running another clinical trial. So during the time that I was doing that MRI work, we were working on all this, and this is a trial done with one of our collaborators, Duncan Stewart, who's at the Ottawa Heart Research Institute, and this is called the Angiogenic Cell Therapy in Acute Myocardial Infarction trial. It's a long name, but essentially it's patients who've come in and unfortunately they've blocked off an artery to their heart. Okay, so they've suffered a large heart attack. We've done everything we can to improve their heart function, but these patients are at very high risk of death or heart failure. So now we're taking those patients, we remove their own stem cells. So we just put a little intravenous in, we wash their blood for a few hours, and we're able to take their own stem cells. We culture them up in a petri dish, and we're able to play around with them a little bit and supercharge them so they're much more effective again, because many patients who have the heart attacks are older and their stem cells age. So we're able to reverse some of those changes, and we're injecting them back. And we've studied over 50 patients so far, so we'll see whether or not this works. And this is, again, been sort of a 10-year project. So as you'll see, all these projects are very long, but I think most things that are worth doing take some time. So we're trying to come up with a catchy title for our program. As you can tell, we've talked a lot about scarring. So, you know, my son, who's in grade 11, loves the old Star Wars. So we came up Scar Wars, the Hardcore Menace, just to, you know, try and keep people interested, keep it catchy. But that's really all I wanted to talk about today. There's a lot of work there, and the important thing is to acknowledge all the people that did the work. I'm the one up here chatting to you guys, but I can tell you that, you know, there's really at least 100 different people that have been working on various parts of these projects for many years. And science is an iterative process. You start off with one and you continually add on slowly over time. And this is the end result of many people's work, and there's a number of people in my lab here. And this is the bridge that connects the research facility to the clinical facility over the road. So it's supposed to represent the bridge of communication and flow of knowledge from experimental work here, and hopefully getting that stuff into clinical trials with humans, and hopefully coming up with the next big advance forward in clinical medicine and preventing heart failure, which is what I'm hopefully in my lifetime. Let's hope that we start to move that bar from 25% death rate at 1 year, hopefully down to 10% or 5% or something much less scary to our patients. So with that, I'll stop. Thanks for your attention, and happy to take any questions.